Hello everyone, welcome back to another video. Today I'll be explaining the regulation of gastric acid secretion. This is a follow-up video from my previous one, the physiology of gastric acid secretion, part of my series of medical tutorials. If you're interested in that, please go have a look. And now back to this video. The secretion of gastric acid is highly regulated by both endocrine and nervous signals. This means there are a lot of hormones involved within the stimulation of the secretion of gastric acid. This includes histamine, gastrin, and acetylcholine. So in this diagram, I've drawn the parietal cells, the lumen of the canaliculi, and the interstitial space. The main objective of the parietal cell is to secrete hydrochloric acid into the lumen of the canaliculi, but how does this occur? It is important to note that this process occurs within the body of the stomach. Within the oxyntic glands, the parietal cells that secretes hydrochloric acid works closely with the ECL cells, also known as enterochromaffin-like cells. And the function of these ECL cells is to secrete histamine. Histamine then binds to H2 receptors on the parietal cells, stimulating hydrochloric acid secretion. In the antral portion of the stomach, there are pyloric glands that contains G cells. G cells secretes the hormone gastrin. Gastrin is a large polypeptide that comes in two forms, G34 and G17. G17 is more abundant, but both are important. Gastrin is then secreted into the blood in response to the digestion of proteins, which is then transported to the ECL cells and the parietal cells in the body of the stomach, in which it binds to CCKB receptors. This process stimulates the secretion of histamine, which will eventually stimulate the secretion of hydrochloric acid. However, it can also directly bind to parietal cells, which also stimulates the secretion of hydrochloric acid. Lastly, we have acetylcholine, which is the chief neurotransmitter of the parasympathetic nervous system. It can bind to M3 receptors, muscarinic receptors, on both parietal cells and the ECL cells. This can stimulate the secretion of histamine from ECL cells, which will eventually lead to hydrochloric acid release. It can also bind directly to parietal cells, which stimulates the secretion of hydrochloric acid. Due to these three neurotransmitters, the proton pump hydrogen potassium ATPase can be activated, which is the driving force of hydrochloric acid secretion. Within the regulatory process, there are always stimulants and inhibitors. There are three main inhibitors of the gastric acid secretion: the reverse enterogastric reflex, the hormone somatostatin, and cholecystokinin. The reverse enterogastric reflex is initiated by the presence of food in the small intestine. This distends the small bowel, which sends an impulse via the myenteric nervous system and vagus nerve, which inhibits acid secretion in the body of the stomach. Somatostatin, also known as GHIH, the growth hormone inhibiting hormone, also inhibits gastric acid secretion. It antagonizes acid production by inhibiting the enzyme adenylate cyclase, which converts ATP to cyclic AMP, CAMP. By antagonizing the G protein receptors, this leads to a reduction in gastrin production by G cells, causing a decrease in hydrochloric acid secretion by parietal cells. Last but not least, cholecystokinin. It acts as a competitive antagonist to CCK1 receptors on G cells and parietal cells. Similar to somatostatin, this leads to a decrease in gastrin secretion by G cells. Cholecystokinin is synthesized by I cells in the small intestine, as well as stimulated by the presence of fatty acid and protein entering the duodenum from the stomach. If you have any questions, please drop it in the comment section down below. Don't forget to like and subscribe. Have a good day.